Hello, everybody. So this is the third video lecture on the second chapter of child growth and development. We left off the previous lecture talking about genetic disorders. Remember that genetic disorders are disordered conditions that are passed along on the genes. And in this slide, in the previous lecture, I was trying to explain the difference between recessive disorders and dominant disorders and sex-linked disorders. Sex-linked disorders, and this is probably more relevant for our conversations moving forward, are the disorders that are passed along on the X chromosome. Remember that your mama has two X's, your dad only has one. So this is why we see, and we'll see this pattern continue, this is why we see more disorders amongst men or present amongst men. Women are likely to be the carriers. So like I say, sometimes jokingly, you can blame your mother. Women are the carriers, but men will be the individuals that present with the disorders. This slide represents a group of genetic disorders. Again, these are on the genes, but these are specifically chromosomal abnormalities. So remembering back a couple slides ago, we had, well, here, I'll just show you. A couple slides ago, we had this image to represent the 23 pairs of chromosomes. The last pair of chromosomes determines your sexual genitalia, your anatomy. You have X and Y, and every pair is the same except for that last pair. But there are some things that can go wrong in this last pair. Well, not just in this last, but a couple of them. So let's start with talking about the genetic chromosomal disorders that affect sex, the sex of your body. One of them is something called a Turner syndrome. And a Turner syndrome is, according to the way it's described in your textbook, is when in development, there were two. There was an X and an X and an X, or an X and a Y, but one of those seems to have disappeared. So you end up with something like an X and an O. And that's what this individual over here is represented as, a Turner syndrome. And it sort of describes some of the physical characteristics. Typically, an individual with Turner syndrome presents, or they appear female, but they are not, but they are infertile. Then you have another one, and this is represented by this Klinefelter syndrome here, and this is somebody that's got two X's and a Y, and the disorder is the additional X. You have another phenomena, sometimes called the supercriminal gene, and that's where instead of two X's, you have two Y's. So that's an XYY syndrome. My understanding is this individual is usually bigger in stature. Sometimes, very often, you'll see cognitive. Remember, cognitive is thinking, so you'll see some cognitive impairments. I know XO is infertile. I believe Klinefelter is infertile. I don't think the XYY is infertile. And triple X syndrome, I believe triple X syndrome is also infertile. And then another genetic disorder that you might be familiar with is the Down syndrome. And this is when instead of it being disordered on that last 23 chromosome, an individual actually has an extra 21 chromosome. So again, this isn't a genetics class, but this would be the 21st chromosome. So you would have two of these instead of just one. So the point is, and one of the things that I think is, I realize as I think about this stuff, is there are lots of things that can go wrong in the process of simply the sperm and the egg meeting. It's amazing any of us are ever born. And we'll actually see that a little bit more as we talk about these different stages of development. For instance, in this very first stage, only about half of the time when a sperm and an egg meet, do they actually survive to this for the first 14 days. So there could have been a whole bunch of times in a woman's life that the sperm and the egg meet, but only half of the time do they ever even get to implantation. This first stage of development, from conception, from the egg and the sperm meeting, to when what they call your zygote, right, from when the zygote implants into the uterine wall, is 14 days, approximately 14 days. It's considered a sensitive period. It's a very, very fragile. This is what we call the germinal period of development. So it lasts for about 14 days, about 2 weeks. After 2 weeks, when we get to the third week, this is what's referred to as the embryonic stage. So this is when the sperm and the egg, they have joined, they've implanted into the uterine wall, and we start to see the differentiation of cells. So we start to see sort of some symbols, some of the early structures of the organs. And there's some really cool imagery on the next slide about how all living creatures kind of sort of start off the same. A couple really important terms here that I hope in the PowerPoint slides that you have are correct is this one right here, cephalocaudal and proxima distal. And what these terms refer to is the pattern of development. So human development, the organs and the systems develop in basically two kinds of patterns. One of them is illustrated here, and this is the cephalocaudal. If you think about your biology classes where you learned about octopus, that are called cephalopods, and cephalopods are the creatures that have very big heads. That's how I remember this. Cephala refers to head. So cephalocaudal development is from the head down. The head develops first. The structures further away from the head will develop later. That is one pattern of development. Another pattern of development is the proxima distal. And the way I remember this is I think about the distal fin on a fish. The distal fin on a fish is on the back, right? So proxima distal development is the midline out. The structures inside the midline, the heart, the organs, those structures develop before the fingers and the toes. So we develop from the head down and the midline out. Those are the structures that happen first, right? Two kinds of development. This stage is what we would call the embryo. It goes on until you get to about 9 weeks. And then after 9 weeks, you're considered a fetus. And fetal development is, by the stage of fetal development, you have all of the major structures. So this is kind of what I was illustrating up here. Oh, this isn't a comparison image, but it is so intriguing that even in an egg, even in a chicken egg, the structures are very similar. So you move to fetal development, right? This is from about week 9 on to birth. You have all of the major structures. When you get to about week 24, this is considered the age of viability. The age of viability means that a person could potentially survive outside the womb at 24. However, as made reference to in your textbook, this is a stage where practitioners, physicians, might not be reluctant. They would be quite reluctant to resuscitate a baby if they haven't made it to 24 weeks. You remember, pregnancy is 40 weeks. So you're barely past the halfway point here. And that's, what does it say? 20, I can't hardly read these pictures, but somewhere in here. And then in the last 2 months, so you've got the last 8 weeks of life, or 8 weeks pre-birth. This is described as a time when the baby is getting ready for birth. So they're putting on weight, they're adding layers of fat, their lungs are developing, things that we're sort of fine-tuning, by the body. And then 37 weeks, is the unborn is able to live outside the womb. My baby was born at 36 weeks, and he was not technically considered, well, he was premature, or because he was less than 40 weeks. They were waiting for that stage because it was considered full term-ish. Okay, I'm going to stop right here at 9 minutes, and when I come back, I will talk about pterogens and some of the factors that make pregnancy outcomes more risky.